Thank you. Well, thank you very much. And the next speaker is Uri Gophna. Uri is a professor at Tel Aviv University. And he will talk to us about from halophilic viruses to antibiotic resistance. I want to say that Uri is really an expert in microbial evolution, and he is one of the top scientists working on Archaea that were mentioned several times in this meeting. I don't know if you're going to talk about Archaea, but if not, you can ask him later. So thank you.
Thank you, Eliora. I think this is my talk. No worries, I have it. No, I will be static. So first of all, I apologize. I'm getting over a cold, so if my voice is a little bad, this is what it is. And I will tell you a little bit about halophilic viruses, really just a taste, and then we will move on to more traditional microbiome work. Okay, so our lab studies microbiomes. It studies, as Noit has introduced, in skin health, mostly in the microbiome in inflammatory bowel disease, and the other part of the lab that does microbial evolution, where actually most of our funding is coming from, is studying selfish elements and horizontal gene transfer in Archaea. And I will just briefly like to mention to those of you who do not know that Archaea are a distinct domain of life. They are unicellular like bacteria, but are genetically closer to us on the molecular level than they are to bacteria, so looks can be misleading, and they have this wide variety of very interesting viruses that are the most diverse in terms of biology in the natural world. So the viruses that infect Archaea are actually very different, and they encompass a broad range of biochemistry and molecular biology, unlike any other viruses in the world, including all those that infect eukaryotes and bacteria. And this is why they are very interesting to study, and they probably have a lot of potentially interesting enzymes that could be used for future biotechnology as well. And Uri Neri, a graduate student in my lab, is now actively discovering such viruses. And to discover viruses of Archaea, you really have to go to environments where Archaea are dominant. And this is actually not the case with most of Earth's biomes. But the Dead Sea is one such environment where Archaea actually dominate. They are, as you can see here, over 80% of the cells in the Dead Sea are archaeal, and it's an ideal place to study archaeal viruses and discover new ones. And here are a few examples of 10 photos of archaeal viruses. You can see that they, oops, I don't know, okay, I'll just point with, sorry. Is there a pointer? Is it the middle? Middle? Okay, so you can see that they have different shapes. So you see this shape, which is like a bacteriophage, and then this very strange shape, and these lemons and spheres, and with these spikes coming out, totally unlike any other virus that you've ever seen. And here you can see a few lemon-shaped viruses infecting an archaeal cell, and this is really a very big playground for us to isolate novel archaeal viruses, both using metagenomics, also using traditional cultivation approaches, and combining the two in sequencing, using metagenomic sequencing of enrichment cultures in the lab. And we are really getting started here, and I hope that I will be able to share more in 2 years' time in a Dead Sea meeting someplace. And this has all been performed by Uri Neri, who is sitting here, who is a wonderfully talented graduate student, and I always say he is a postdoc level graduate student, although he's only in his second years of master's. So if you have great students, you can make very big leaps of science, even if you don't have a very big lab. Okay, but now to other extreme. So extreme, in this case, would be long antibiotic exposure, and what extremely bad things it does, but sometimes also good to gut microbiomes. And our model system is this. In this case, it's ileal pouch anal anastomosis, or as it is more often called pouch surgery. And this is a surgery that is performed in patients with severe ulcerative colitis, so a form of inflammatory bowel disease that does not respond to other treatments. So unfortunately, these patients sometimes have to undergo complete proctocolectomy, which means that the entire large bowel is removed, and to allow them to lead normal lives, the pouch is formed out of the terminal part of their small bowel. This is the thing you see there in the corner, and this allows them to have normal bowel function. The problem is that the majority of these patients, after having undergone this surgery, will then develop inflammation of the pouch. It is called pouchitis. And when they do develop this, antibiotics is the first line of treatment and generally works well, but some patients actually become antibiotic dependent. So they need to take antibiotics constantly to maintain their remission. And when they stop taking the antibiotics, then in a month or two, they will relapse and have pouch inflammation again. And to us, as microbiologists, this does not make any sense, because if you treat these patients, end up being treated for months and years with a cocktail of antibiotics, and it would not make much sense, because you'd think that bacteria would sooner or later become resistant, right? Even to a non-microbiologist in the audience, I'm sure that this is immediately obvious. So this is why you are not gastroenterologists. And so we asked ourselves this question: what are the prevalent antibiotic resistance mechanisms in these pouch microbiomes? What part of the microbial community becomes resistant? Is it that some bacteria are resistant and protecting others? Is there collateral resistance? So if we treat with these drugs, do we also make the bacteria in these microbiomes more resistant to other antibiotics? And then finally, why do patients become antibiotic dependent? So why can't they stop the treatment once they are on it for a long period of time? And just to go over it briefly, so we have for us what is considered a fairly large sample, so it's 40 patients, which is really a lot in this particular disease, and over 200 fecal samples taken longitudinally over several years, and most of these patients have had a long follow-up and a lot of treatment during these years, and generally treated with a combination of an antibiotic of a fluoroquinolone called ciprofloxacin and metronidazole, and usually both of them together. And then we do, like we just heard, metagenomic sequencing. So we sequence everything so we can determine not just the taxonomy, but the gene functions, and especially resistome functions. So genes or alleles that can confer antibiotic resistance, we can then predict from the sequence data. And this is all done by Vadim Dubinsky, a PhD student in our lab. So first, what are the prevalent antibiotic resistance mechanisms in these pouch microbiomes? So fluoroquinolones, they target DNA gyrase and topoisomerase. So for those of you not from molecular biology, these are enzymes that have to do with the supercoiling of the bacterial DNA. So whenever the bacterium replicates its DNA, if these are inhibited, breaks will appear and the bacterial cell will die. And these are broad spectrum, have been used very successfully since the 1980s. And so the targets are the DNA gyrase, the DNA topoisomerase, and you could have resistance if these targets are mutated. So there are point mutations in these proteins that then make these proteins insensitive to the drug because the drug can no longer bind to them, and this can appear just by point mutation and have this, and can confer the strongest resistance. There are enzymes that can modify the drug, and these are often located on mobile genetic elements such as plasmids, and for example acetyltransferases, and there are target protection proteins, so proteins that can bind to the topoisomerase or the gyrase and then prevent the drug from binding to it. And these are called QNR genes, and they are again mobile, so found on plasmids, on mobile DNA that can be transferred across bacteria. So when we look at our metagenomes, what do we find? We find that pretty much in all the metagenomes taken from patients treated with antibiotics, we see point mutations that confer resistance to the drug. And often we see 3 resistance mutations, and if we see all 3 mutations in one genotype, in one species, that means that this one would be totally insensitive to the drug. So this corresponds to total clinical resistance to the drug. We also see a little bit of mobile resistance, but to a lower degree. So less than a quarter of the samples had the acetyltransferases, and less than 15% had the QNR gene. So most of the resistance that we see is actually chromosomal and not mobile. Then if we do see how much of the community becomes resistant. So because we know which chromosomal mutations confer resistance, we can then map the reads to the sensitive alleles, and we can see where we find mutations that are supposed to provide a level of resistance, and we can then do the same thing for every species. So here it's illustrated for E. coli, which is an easy example. If there is a C and a G, and all the reads map to it, so you see all the reads are gray, which means everything is identical to what we see on top, then this is all the sensitive alleles. So this is from a control sample taken from a patient not treated with antibiotics. And here you see all the E. coli strains in this patient were sensitive to fluoroquinolones. And this is what happens when you have a patient that was treated with fluoroquinolone and then for a long period of time, and this is a sample taken during antibiotic treatment. So you see here the two resistance mutations appear together on the same read. And this means that this E. coli, all the E. coli sequences that we see from that patient are actually fluoroquinolone resistant. And if we look at the patient shortly after beginning antibiotic treatment, so we still see a little bit of the sensitive allele, so here and there, but already 2/3 of the E. coli strains that patient has have become antibiotic resistant. So this gives us some idea on how fast the antibiotic resistant strains take control of the microbiome. And we can do this then for every genus or every species in the microbiomes of these patients, and then we can then assign these sequences to being either resistant or sensitive. And here we labeled the same groups of bacteria, in this case order level, but it doesn't matter. We gave them warm colors if they're resistant, and then cold colors if they are sensitive, and if it's white it means they are absent. And on the top you have samples taken from antibiotic treated patients, and on the bottom you have samples taken from patients that were not treated with antibiotics, and you could already tell that the vast majority of the microbiomes of patients treated with antibiotics have become antibiotic resistant due to mutation, and this is not so much present, of course, as we expect in those not treated with antibiotics. And if we summarize, we can see that overall about 70% of the microbiome of the antibiotic-treated patients has become antibiotic resistant, and this is the background resistance is lower than 10% in patients that have not been treated with antibiotic. And we also see a community-level shift from the bacteria that you normally find in gut microbiomes to one that is more dominated by Escherichia coli and Enterococcus that are antibiotic resistant that we find in the treated patients. So is there collateral resistance to non-related antibiotics? So these patients were treated with a cocktail of 2 antibiotics, and mostly what is having an effect is the ciprofloxacin, this fluoroquinolone. Will we see also resistance to non-related drugs that these patients have not seen during their treatment? And the sad fact is that we do. So if we just quantify how many of the genes that we sequence using shotgun metagenomics are antibiotic resistance genes, and we quantify the fraction of genes in antibiotic treated and non-treated patients, we see about 2-fold increase in antibiotic resistance to genes that have nothing to do with the antibiotic that these patients were exposed to in the antibiotic treated patients. And if we then stratify by the time since antibiotic treatment, we can see that this effect gradually goes away the longer you are for the treatment. So if you've stopped being treated with antibiotics for over half a year, then you already look like antibiotic treatment naive, and you no longer have this increased fraction of resistance genes in your metagenome. And the reason for that increase in resistance to non-related antibiotics is that some fluoroquinolone resistance genes, some genes that confer resistance to the drugs that these patients have been taking, are actually localized on resistance plasmids. So, and then on the same contig, in the same stretch of DNA, we already sequence fluoroquinolone resistance genes. We see genes that confer resistance to other antibiotics, because genes to antibiotic resistance tend to occur on the same cluster. This is a well-known phenomenon. So for example, here we see a lot of resistance to beta-lactams and cephalosporins. We have extended spectrum beta-lactamases that are encoded on the same elements as mobile fluoroquinolone resistance genes. So this is really not such good news for these patients. They are being treated with one antibiotic, but actually they harbor resistant bacteria to many other antibiotics as a consequence. Finally, a question that we are often asked is if we are enriching for these bad bacteria, why do patients respond, right? You're taking away their good microbiomes. You're giving them now enriching for E. coli, Enterococcus. They are considered to be bad bacteria, especially for IBD patients. But actually, if we look at the gene level, we see that we can map the genes and find out if they have genes that are important for intestinal inflammation, so ones that are predicted to damage the epithelium of the intestine or to increase permeability. And actually, when we do this, we see that they are absent or virtually absent from those Archaea and Enterococcus strains that these patients have in their microbiome. So this explains may have bad species, but at least they have lucked out and they have resistant but non-intestinal pathogenic strains, which is why they keep responding to the treatment. In contrast, when we look at patients when they stop their antibiotic treatment, we sometimes detect strains that have quite potentially intestinal virulence genes, and we also see high inflammation scores for those patients during those time points. For example, we see this enterotoxin of Enterococcus present in 5 samples from 3 patients when they stop taking antibiotics. So sometimes when you have an antibiotic-resistant strain, if it's virulent, this is the one you want to keep. So why do patients become antibiotic-dependent? As I said, many of these patients, when they stop taking their antibiotics, they are okay for a month, and then they relapse, and they have to take antibiotics again. And then we try to look at what happens to them when they stop. And what we see when they stop is something like this. So here, this time point, that patient was taking antibiotic here as well, and here they stop taking their antibiotics, and you see an increase in a very strange set of species. So these can be either oral bacteria, so bacteria that are generally not found in large amounts in the intestine. They are only found in the oral cavity. And we also see some things that don't belong, like an avian Enterococcus species that is normally only found in chickens, not in humans. And we also see these that are labeled pathobionts. So these are species that may be okay if they are in you or I, but they have been associated with inflammatory bowel disease in 2 or more human studies. And these seem to increase once antibiotics are stopped. And we then quantified this, and we saw that actually there is a big increase in these atypical bacteria when antibiotics are stopped for patients that have been treated with antibiotics in the past. And we take this to mean that these patients that have been treated with antibiotics for long periods of time actually now rely on antibiotics for their colonization resistance. So when they stop taking the antibiotics, everything can colonize, and the bacteria that colonize tend to be rapid-growing, fast colonizers, and these sometimes are pro-inflammatory. So just to conclude, antibiotic-resistant strains have low virulence potential, which is why probably the treatment works. Most resistance is by point mutation and non-mobile, but there is a big but. And most of the community becomes antibiotic resistance, and the colonization resistance it confers is also antibiotic dependent. So when the antibiotics are stopped, then bacteria that are fitter then take the place of those antibiotic resistant bacteria that have been selected for probably mostly by the treatment. And finally, the antibiotic treatment, while it mostly selects for point mutation, it also results in collateral resistance against other antibiotics and enriches for strains that carry plasmids that carry multi-drug resistance. So this treatment actually generates a long-term risk for patients and their family. So just to thank the people who did the work, so most of the work was done by Vadim Dubinsky in our lab, head by Dr. Lea Reshef. We would also like to thank the patients for volunteering for our study, and of course our clinical collaborators, the Pouch Clinic, Dr. Iris Dotan and Dr. Hagit Tulchinsky, and the IBD lab members at Rabin Medical Centers, and the Helmsley for funding. And thank you for your attention.
Thank you, Ori. This was was also a good thing you didn't talk before lunch.
Were you able to, or do you have any indication for protective bacteria against the inflammation?
Well, this is not the best setup to find protective bacteria, but actually we know that one of the problems of antibiotic treatment in pouchitis, that on the one hand it's a very effective treatment, but it virtually destroys all the bacteria that are thought to be protective in IBD. So it is really a double-edged sword, even if we take out of the equation the part of antibiotic resistance.
If I may, I have a question both for you and for our previous speaker. As a physician, I'm being yelled at about washing my hands. But it's not just washing my hands. It's now washing my hands with an antibiotic something or other, a gel. And I'm wondering if we're setting ourselves up for trouble by doing this, by developing antibiotic-resistant organisms on our skin, which in the olden days you'd get rid of by washing your hands with soap and water. Is it possible that we are digging ourselves a hole that's deeper than the one we already have?
Well, all I can say is that in the US, antibacterial soap has been for many decades the thing, and people would use antibacterial soap at home. In Israel and in Europe, most of the time you only use antiseptic soap in hospitals, where it's actually, I think, better justified. So what people have shown in the US, that due to the use of antibacterial soaps and also toothpaste, the bacteria that people have are already resistant to those antiseptics and to, I forgot the name of the molecule that's found in toothpaste in the US. But this is probably, my take on it would be that antiseptic soap should only be used by medical staff and people in hospitals, but not by people at home. So the question was, should we not do a fecal transplant right after they stop the antibiotics? And we think we should. And people we were a little bit worried about doing fecal transplant with pouch patients because the volume of the pouch is so much smaller than a colon. So this is like a 200 milliliter at most. It's a very small organ compared to a full colon, and we were a little bit afraid, but people have done pilot fecal material transplant studies in the US, and it seems to be that it is safe for these patients. So we should consider it, and this is one of the final lines of the discussion in our manuscript. So we think this is true. This is the way to go.
I had a question about your kind of bystander effect.
Wait, so everybody will hear.
I can repeat the question.
My question was about your bystander effect. I think we use antibiotics and we believe it has an effect on the bacteria we treat. But in fact, we may have as a target all the 10 to 14 bacteria that George described in our gut microbiome. So in general, I mean, I've not seen a lot of studies looking when you give an antibiotic, whether you look at whether the resistance is by bystander effect is transferred to many other bacteria, and what happens to them in the long term. How much the resistome, if you like, is accumulating in our gut.
So I agree that this has been comparatively little studied, and this is why I was really happy that I had multiple time points from the same patients. And also I was happy to see that a lot of the effect goes away after treatment cessation, because I was afraid that some of it will stick. I think people should study this in more, let's say, standard individuals with colons. And I think this is something that should be done. Again, it's something that we are writing in a paper right now as a recommendation for future study.
So just to add a little bit of that, Rito, in the athlete microbiome project, every now and then somebody would break a finger, they would bang into a tree or something, and they would keep racing for the rest of the season, but they'd be given some antibiotic. So we had an opportunity in a longitudinal study to look at the effect on very healthy people, hit them with an antibiotic, and follow it over time. And these were different antibiotics. It's sort of all over the place. Some of them would get, beta-lactam or something like that, along with something to inhibit the beta-lactamase, and it would just wipe out everything in their gut except Bacteroides or something like that, and then it would take a long time to come back. Others would get something, and it would just make little quantitative effects on things. So it seems like it's very difficult to see. Marty Blazer is leading a project called the MIME project, which is looking at multiple microbiome sites in healthy people who will volunteer and give antibiotics over a year. So you can really look at everything. So stay tuned. There'll be a much more comprehensive, I think, experimental design for this. But it's still, I think, very much of an unknown, other than the fact that it makes a big change and it takes a long time to come back.
Thank you very much, Uri, and thank you for